Now, a few years back, I was visited by some executives of the Christian Dior company, and I was a little suspicious. What do I know that Christian Dior would like to get involved in? And as you can guess, they have a commercial product. Their chemists have identified some small molecules that lead to a subtle increase in the expression of aquaglyceroporin 3 in skin. Now, I should point out, I have no financial ties to Christian Dior. In fact, I would love to have financial ties to Christian Dior, but none were offered under conditions I could accept. So they're marketing something called hydroxyl therapy. It's quite expensive, about $50 for a 3-ounce jar. And this is now the beauty industry. This is not drug development. So I think the implication that is if you use enough of this, you'll look like this. I'm not sure. I don't think they've really proved that. But this was the back cover of the Marie Claire magazine, which I purloined from the Charles de Gaulle Business Lounge. And those of you who read French will see some very bold statements. Profound hydration, spectacular results. Oh, what's this, the Nobel Prize in chemistry? I showed this to my mother, and she smiled and said, "Peter, you're finally doing something useful." Bridging science and society, I guess. I thought the other stuff was pretty useful, but you know, our mothers really know us better than we think. But the aquaglyceroporin proteins probably are important in a number of clinical states, and I'll just tell you about a few. First off, they're present in red cells, and explain the glycerol transport in red cells. But they might be particularly important in the clinical problems of malaria. Malaria being a very important disease affecting particularly children in the third world. And it goes through a complex life cycle, but at one point there's an intracellular parasite which rapidly grows and divides, devouring hemoglobin, breaking, and causing tremendous toxic fevers. Shown here is peripheral blood of Plasmodium falciparum, and here is the initial ring stage, and here's what they call a schizont. So this was rapidly divided, filling the red cell, and causing tremendous clinical hardship. And it turns out the glycerol transport mechanisms do have a partial effect. We'd hoped they would be more important in the infectivity of this organism and might be drug targets, but you live and learn in science. We're now working at the aquaporins in this critter, Anopheles gambiae, the mosquito that transmits malaria and contains 6 different genes encoding aquaporins. And there's one obvious problem that the Anopheles have, and that's during the blood meal, when it will feast on humans taking up blood infected with malaria. And in order to take up enough blood to make eggs, it must rapidly move fluid through the Malpighian tubules. So this might also be a target for small molecular approaches to infecting the mosquitoes carrying malaria. We'll find out soon. Just a word about the clinical states of malaria. This is a heartbreaking disease, causing more than 1 million deaths per year, largely of small children. And these little boys, these cute little fellows, they live in the huts and in the hamlets outside of our malaria station in Zambia. And while they look perfectly happy and healthy, they've each had malaria a number of times. All children in rural Zambia have had malaria in the past. But with rapid diagnosis and good medical treatment, they've recovered well. And by the advances of public health, with bed nets, residual screening, the malaria incidence in some parts of this country are declining. But it still is affecting some children. And in addition to the deaths, there are many more children who are affected with malaria who survive, but they survive greatly impaired. Shown here is a little child who was brought into the clinic at Macha, Zambia, where our colleagues are working, nearly dead in coma. He was rescued, but if you can look closely at those in the front, at least you can see that he has a disconjugate gaze. He's left with cortical blindness, which he'll probably never recover from. So there's a lot of reason to work on this disease, and the medicines that are effective against malaria, unfortunately, are declining use because drug resistance is now emerging. So we have a big challenge on our hands. I'll tell you briefly about 2 other members of the aquaglyceroporin family. Aquaglyceroporin 7, which is present in adipose tissue, 9 expressed in liver. Now, we have a new disease. It's not a new disease, but we have a new epidemic in the United States, and that's called obesity. Our citizenry are eating too many calories, and because of the profound evolutionary stress with starvation, humans are very adept at storing these calories as triglycerides and fat. Now, during fasting, the triglycerides are degraded, and the preferred gluconeogenic substrate, glycerol, is released from fat tissue through aquaglyceroporin 7, and it's taken up by the liver. And the liver expresses aquaglyceroporin 9 at modest levels unless fasting occurs, in which the levels rise dramatically. So glycerol taken up by the liver converted to glucose. And this is work of Jen Carberry, who South Carolina young lady who's now a faculty member at Duke. Now, a very interesting surprise, and this is not rare in science to find unexpected results. And these results emerged from the work of Barry Rosen and his team at Wayne State University, who were studying arsenic toxicity using yeast as a model system. And what they discovered is that these aquaglyceroporin 7 and 9 were freely permeated by arsenite. So the arsenite-resistant yeast could be made sensitive to arsenite. Now, arsenic in neutral pH exists as arsenic trioxide. It's uncharged and probably chemically resembles glycerol. What is the significance of this? Well, it turns out that in some parts of the world, fortunately not the United States, but unfortunately Bangladesh and eastern India, the groundwater is contaminated with cholera. And so the government, with limited resources, put in tube wells, shown here, to draw, the surface water is contaminated with cholera. The groundwater is then tapped by the tube wells. But a lot of these water sources are contaminated with huge levels of arsenic. And the current indications are that 140 million people in that part of the world are drinking arsenic-toxic water every day. Now, in a recent series of studies with knockout mice lacking with targeted disruption of the aquaglyceroporin 9 gene, it turns out that the aquaglyceroporin 9 is probably a detoxifying mechanism allowing the excretion of arsenic through the liver. Now, all of this is very interesting and academic, but how does it help people? And in fact, in this instance, it can't help people because the prevention of arsenic toxicity is the provision of pure drinking water, which is a basic human right. Now, I'm working at a medical school now, a school of public health, focused largely on human biology, but others working in plants have had a field day studying aquaporins in plants, including here at UC San Diego, and I see him here, my friend Martin Crispiels. And together, Crispiels and his colleagues have identified many water pathways through plants, which are explained by these aquaporins. And one slide I borrowed from Ralph Kaldenhoff from Germany shows two Arabidopsis plants. On the right, a parental plant, on the left, a genetically modified plant, modified to underexpress the rootlet aquaporins. And as you can see, that plant has compensated. The foliage looks the same, but below the surface, you can see an increased arborization of rootlets. And these plants have approximately 35 different aquaporin genes. The rice genome has 50. So plants are very busily using these molecules to move water through their tissues. So I've told you about a lot of studies, mostly from our laboratory. There are quite a few other laboratories involved. But I'd just like to show a photo of some of the young people here, because science really is about young people. I was once young, I'm not any longer. But this wonderful endeavor we have that includes young scientists working in laboratories leads to major advances. And they've come from all over the world to join us in our laboratory, and each has contributed to this study.